The next type of oxidation that we are going to see about is the alpha oxidation. Alpha oxidation and omega oxidation do not require the coenzyme A intermediates. Since there are no coenzyme A intermediates, there is going to be no generation of energy as well. In alpha oxidation, there is going to be removal of a carbon atom from the carboxylic side in the form of carbon dioxide. So, only one carbon atom is going to be removed sequentially in alpha oxidation. The reactions of alpha oxidation are going to take place in brain and other nervous tissues. Let us see about the reactions of alpha oxidation one by one. The first reaction in the alpha oxidation is the action of monooxygenase, which is going to act in the presence of oxygen, vitamin C and Fe2+. Now, there is going to be an introduction of the hydroxyl group at the alpha carbon atom of the fatty acid. Then this fatty acid, which now has an alpha hydroxy group, is going to undergo an oxidation reduction reaction in the presence of dehydrogenase enzyme. Now, the alpha carbon atom is going to have a keto group in the form of C double bond O. Now, this alpha keto fatty acid is going to be acted upon by the decarboxylase enzyme, which decarboxylates this alpha keto fatty acid to form carbon dioxide and the fatty acid, which is now having one carbon less. Role of alpha oxidation. Alpha oxidation is basically the oxidative decarboxylation of the long chain fatty acids. Now, because of this oxidative decarboxylation reaction, the even chain fatty acids can be converted to odd chain fatty acids, which are constituents of the complex lipids of brain. Phytanic acid, which is a 20 carbon fatty acid, also a principal fatty acid from the chlorophyll, is usually oxidized with means of the alpha oxidation. Disorders of alpha oxidation. Refsum's disease. Refsum's disease is also called as phytanic acid storage disorder. It is a rare neurological inborn error of lipid metabolism. There is going to be a deficiency of phytanic acid alpha hydroxylase, which is required for the alpha oxidation of phytanic acid. This phytanic acid is usually derived from the dietary chlorophyll. Now, phytanic acid cannot undergo beta oxidation because of the presence of a methyl group at carbon number 3. This methyl group is usually removed by alpha oxidation. So, when alpha oxidation is impaired, there is going to be an accumulation of the phytanic acid, which is going to cause symptoms such as retinitis pigmentosa, peripheral neuropathy and ataxia. The treatment of this disorder is to simply eliminate the dietary sources of phytol. The next type of oxidation that we are going to see about is the omega oxidation. Fatty acids may be oxidized at the omega carbon of the chain by the enzymes in the endoplasmic reticulum. Now, the omega carbon is the carbon that is farthest from the carboxyl end. Short chain dicarboxylic acids such as pymalic acid, which is a precursor for biotin, is formed from the omega oxidation. Reactions of omega oxidation. Omega oxidation involves the conversion of fatty acid into dicarboxylic acid in a two-step reaction, which is usually catalyzed by the cytochrome P450 hydroxylase enzyme. This enzyme requires the presence of oxygen and NADP. In the first step of the reaction, there is an introduction of the hydroxyl group at the omega carbon atom, which then rearranges to form the dicarboxylic acid. Dicarboxylic acid that is produced in the omega oxidation undergoes beta oxidation in mitochondria and usually forms adipic acid, which has 6 carbon atoms, and subiuric acid, which has 8 carbon atoms, which are then excreted in the urine.